Hello, everyone. You might think of yourself as one single human being, and psychologists would argue there's nothing wrong about this. However, if you were to ask a biologist, he might disagree. As a matter of fact, we consist of muscles, blood, bones, and more or less hair. And all of these is a collection of cells. Trouble is, if, for instance, you were to look at blood cells, these are very, very tiny. And despite the fact that a blood drop might appear red, if you look at the individual cells themselves, they are transparent. So this means if you look under it, under a microscope, on cells, you won't be able to see anything. This is why biologists have been using for a long time chemical markers to stain, to color the cells. The problem with these chemical markers is you basically kill the cell, so you can only observe dead cells. And the other problem is these colors, they only stick to certain parts of the cells. In this example, you can see the membrane in orange and the nucleus in blue. So every time a researcher would like to see a different part of the cell, he has to use a new marker and a new cell. And besides killing the cell, this staining process might actually alter the state of the cell. So eventually what you observe from the cell might be very different from the original living cell. We heard earlier from Daniel Bora about butterflies. So we can make a comparison with butterflies. Let's assume there was a species which is transparent. The only way to visualize them is therefore to spray them, so to give them some contrast. And now you can see, in this example, only the wings of the butterflies. But only seeing the wings doesn't really help you to understand the system. So wouldn't it be great if you could see the entire butterfly? Or even better, if you could see the butterfly in 3D, measure it in 3D, or the best of all, to see it alive as it moves in its natural environment. And this is the very vision we had for Nanolive. Of course, not with butterflies, but with cells. So the first thing we did, we trashed chemicals. No more chemicals. That's past. Instead, what we use is another physical property of cells, which is called refractive index. So for instance, if you were to shine a laser light on the cell, the refractive index, this optical property within the cell, is different from its environment, and therefore it will deflect the laser beam. And this is something you all know from daily life. For instance, if you put a pencil, as you can see here, you put the pencil into a glass of water, the pencil will appear bent. And based on this physical principle, we created the very first microscope in the world, which enables researchers to see and to interact with cells as they are alive and in 3D. It's called the 3D Cell Explorer, and as you may know from an MRI in hospital, we scan around the sample, and with the help of a computer, we then reconstruct a three-dimensional image of these cells. And talking about applications, this allows you, for instance, for the very first time, really the very, very first time, to visualize then fundamental processes like cell division in 3D and without harming or killing the cell. Another application medical doctors are looking at in cancer therapy is to find the best drugs which will effectively kill the cancer cells. So what they would do, they would expose the cancer cells to different drugs, and within no time, with the Cell Explorer, you can then identify that the yellow drug in this case is the most efficient one. While traditional microscopes, these color microscopes, these staining microscopes, sell at very, very high price points, with Nanolive we had a different vision, and we wanted to make the Cell Explorer at a very affordable price point. So 20K instead of hundreds of thousands. And with this price point, we can enable anyone who wishes to interact with their cells. We can say, yes, you can. And and today we invite you, so have a look at our homepage, nanolive.ch, spread the word, or we brought actually a microscope to our booth. You can come and visit us and have a look for yourself. Thank you very much.